Hi, I'm Jang-Yeon Park from Jeongbyon University in South Korea. And in this talk, I will talk about in vivo direct imaging of neuronal activity at high temporal and spatial resolution. I have nothing to declare. People have been trying to understand how the brain works, especially by trying to understand the brain's functional organization and ultimately including dynamic and hierarchical functional connectivity. In order to do that, people have developed several in vivo non-invasive neuroimaging methods, such as EEG, MEG, and fMRI. And they can provide valuable information on the brain's functional organization, but at the same time, they have obvious pros and cons in terms of temporal and spatial resolution. For example, in the case of EEG and MEG, they provide excellent temporal resolution on the order of milliseconds or less, but they provide relatively poor spatial resolution in centimeter scales, especially in deep brain areas. And in the case of functional MRI, people have been using several different contrast mechanisms, such as BOLD, CBV, and CBF, which are based on the neurovascular coupling between neuron activation and hemodynamic responses. And they can provide excellent spatial resolution on the order of a millimeter or less at ultra-high field over a wide brain coverage area. However, they provide relatively poor temporal resolution on the order of seconds because they are based on hemodynamic effects, and they provide inherently indirect information about neural activation. So there has been a long-standing and challenging question in MRI community, such as, is it possible to directly detect neural activity using MRI, especially in vivo? So the motivation of this study was that what would happen if functional MRI could have ultra-high temporal resolution on the order of milliseconds? This motivation originated from the fact that neural activity is a transient effect which has a milliseconds order timescales. For example, axon potential has a time duration of a couple of milliseconds. So in this case, the basic idea was if we could have milliseconds temporal resolution, we might have a better chance to effectively capture the transient effect of neural activity in milliseconds. And in addition, we could elucidate the hierarchical functional connectivity of the brain by tracking the neural activity propagation. Also, we could reveal spatial temporal dynamics of neuronal networks in multiple functionally connected brain regions or circuits. So in order to implement this idea, I thought of an ultra-fast imaging technique such as EPI, which has been widely used in conventional bold fMRI. Because I had a preconception, like many people, that scan time is a temporal resolution. But even in the case of single slice EPI, I realized we could only achieve tens of milliseconds temporal resolution. However, scan time doesn't have to be the same as temporal resolution. According to the very nice idea of line scanning, first proposed by Dr. Alan Kuretsky's group, we could significantly increase the temporal resolution by acquiring a piece of case space rather than acquiring the entire image at one time. These are the schematic diagrams of data acquisition in conventional bold fMRI and line scanning. In the case of conventional bold fMRI, we acquire first time frame image and then move to the second time frame image and so on. However, in case of line scanning, we acquire only the first case space line of the first time frame image And then we acquire the first case spatial line of second time frame image and so on. And after first phase encoding, and then move the second case spatial line, we do the same thing, and so on. In this case, the repetition time TR would be the same as the temporal resolution. So using this line scan idea, we could implement milliseconds temporal resolution imaging. So we implement the 2D line scan gradient echo imaging with a short TE and short TR. The TE was 2 milliseconds, TR was a 5 millisecond, which is equivalent to the temporal resolution in this case. We performed experiment using mice on a 9.4 Tesla animal scanner. We repeatedly applied the electrical stimulation to mouse whisker-pads with an interstimulus interval of 200 milliseconds. So in this case, if we divide 200 milliseconds by 5 millisecond TL, we see we have a 40 time frame images. Then the spatial resolution was a 220 micrometer in plane resolution and 1 millimeter slide thickness. And this is the first result of imaging neural activity and milliseconds of temporal resolution. From now on, I will call our methods as Diana, which stands for direct imaging of neural activity. In the figure C, you see the time series of Diana t-value maps with the 5 milliseconds temporal resolution in the primary somatosensory cortex of battlefields, which is a S1BF as abbreviation. You see here activation. And in figure D, you can see that there was a statistically significant Diana signal in the range between 0.1 and 0.2% when compared with the control without a stimulation and another control post-mortem with stimulation. To validate this result, we also performed electrophysiological recording with the same stimulation setup. And as you see here, the Diana response, the envelope, had a very good correlation with the stimulation responsive single unit activities. And it is interesting to note that the LFT has a little more delay than Diana response. And now, because we have a milliseconds order temporal resolution, we could track the neural activity propagation, for example, in the thalamocortical pathway in this case. So we investigated the Diana response in thalamus and S1BF, and as you see here in figure D, we could observe the Diana response in the thalamus first between 10 and 15 milliseconds, and then after that is we could observe the Diana response in contralateral S1BF between 20 and 25 milliseconds. And these results were also validated by the electrophysiological recording in both thalamus and contralateral S1BF. Diana response showed a good correlation with the stimulation responsive single unit activities. Then we performed another experiment to show that the Diana response is not a body effect. So we designed two experimental conditions. One is an extra oxygen and another one is air only. So consistent with the previous Boulder studies, you could see that is with extra oxygen, there was a higher Boulder response in both thalamus and contrast MBF. But in contrast, in the case of Diana response, we could barely see the changes in Diana response in both thalamus and contrast MBF. So we could conclude that Diana is barely affected by the Bowley effect. And based on this result, we also investigated why this hemodynamic response is well suppressed in this Diana acquisition. The first reason is the theoretically and by simulation. The first reason is the short echo time of Diana. For example, the echo time was 2 milliseconds in Diana acquisition compared to the 20 to 30 milliseconds of TE in conventional bold fMRI. Another reason is that the characteristics of Diana scan acquisition itself with a relatively short interstimulus interval, for example, 200 milliseconds in this case, is compared with the slowly varying hemodynamic response. The result of simulation is presented in the paper, the supplementary material. Another reason is that with consistent with the previous another reason comes from the previous bold fMRI studies. It's already known that there was a significantly reduced bold response with high frequency, is above 1 Hz stimulation. So for example, bold signal was suppressed up to the 0.021% with a 0.75 Hz stimulation at 7 Tesla. Then what would be the underlying contrast mechanism of Diana? If we are based on the neuronal current model, for example, the neuronal current induced local magnetic field change might not be the candidate because it causes a dephasing of proton spins, and which will lead to the negative signal change. But as we could see in the previous slide, the dyno response has a positive signal change. So this model will not work. So now, at this stage, we have a strong hypothesis that the underlying dyno contrast mechanism may be the T2 change due to the changes in membrane potential during neuron activation. At the biophysical level, this T2 change is may be attributed to the changes in water molecules in hydration layer and cell swelling, both of which are accompanied by the changes in membrane potential. Now, let me explain first how the T2 changes due to the changes in water molecules in hydration layer. So as you see here, in a resting state, let's assume that in the resting state, this is the amount of hydration water, which is known to have a short T2 in the range of between 0.1 and to the 10 milliseconds. In contrast, the free water has a relatively much longer T2 in the range of seconds. So in the case of depolarization, you see here, the hydration layer water will be decreased, which means that the portion of this reduced portion of hydration water will be released to free water, which has the longer T2, and then the T2 of the system will increase, and then the PSR, so one of the MR magnetization transfer parameter in MRI, which is defined as the ratio of bound water protons to free water protons, will decrease. And in the case of hyperpolarization, opposite thing would happen. The amount of hydration water will increase, which means that the short T2 components would increase. Then in overall T2 will decrease, and the PSR would increase. To confirm this hypothesis, we performed the in vitro cell experiment using T-cell lines at 9.4 Tesla. The T-cell lines are immortal and non-excitable cells, which means they are stable in membrane potential and could survive for several hours during the MRI scan. And as you see here, in the case of depolarization, there was about 15% T2 change and about negative 50% in the hyperpolarization. And in the case of PSL, there was a negative 17% change, and also in the depolarization. And in the case of hyperpolarization, there is about 80%, positive 18% change of PSL. that was consistent with our hypothesis. We also performed experiment using another cell line. A different type of cell line is a neuroblastoma cell line, SHS5Y. And you also see that in the case of depolarization, uh, there was about 20% of T2 change. And in the case of hyperpolarization, there's about negative 4.4% of T2 change. And there was a negative change of PSR in the depolarization and positive change of PSR in hyperpolarization, which was also consistent with our hypothesis. To validate this hypothesis in vivo, we also performed in vivo experiment using rat brain at 7 Tesla. First, we pharmaceutically suppressed the hemodynamic effect. And then we measured T2 uh, after craniotomy by perfusing the ion concentration. And there was about 3% of T2 change there. We also measure T2 change due to cell swelling by only changing the osmotic pressure, not membrane potential, using T cell line. There was a non-negligible T2 increase in both intracellular space and extracellular space. Now, I'd like to discuss some important implications of Diana, which are caused by a direct correlation of Diana with neuronal activity. So one of the important implications is that we may need to consider variability of neuron responses seriously in Diana data acquisition and Diana data analysis. For example, if we accept this kind of implication, the averaging more runs may not guarantee better sensitivity of the Diana response. And this is one representative reference that explains the large variability of the evoked neural responses in cortical area by using the concept of spontaneous ongoing activity. As you see here, the evoked activity in the mammalian cortex shows a large variability to the repeated presentation of the same stimulus. And the evoked activity appeared deterministic response, and the neuronal variability resulted from the dynamics of neuronal ongoing activity, which presumably reflects the instantaneous state of cortical networks. And this figure shows two individual responses of optical imaging, local field potential, and single unit activities to repeated visual responses in a 2 square square area of the primary visual cortex of an anesthetized cat. As you see here, with the same stimulus, the result response are different. They have a variable. They shows the variability in optical imaging, local field potential, and single unit activity. Of course, if we average them, they show some kind of a pattern. According to our experience, the sensitivity of Diana response is not very improved by averaging more number of runs, rather degraded often. However, when we average Diana data across the mice, the sensitivity of the Diana response is quite well improved. This is the example of a representative example of Diana raw data from average of 40 runs in a single mouse after pre-processing. As you see here, even if it's very nice Diana raw data, it has a very large fluctuation, baseline fluctuations compared to the Diana response in thalamus and contrast on BF. I hypothesized at this stage that this large baseline fluctuation may come from spontaneous ongoing activity I mentioned in the previous slide. Interestingly, when we average across mice, this large baseline fluctuation is quite well suppressed, as you can see here in figure D. This is the result of average of 10 mice. So according to our observation, the spontaneous ongoing activity, which may be reflected in a large baseline fluctuation of dynamical data, it appears to be reduced significantly by averaging across subject rather than averaging across runs in a single subject. And this is presumably because spontaneous ongoing neural activity seems to be specific to each subject. For example, having its own frequency or phase. If the spontaneous activity represents some neuronal oscillations in brain networks. Based on this observation interpretation, I think we may need a paradigm shift from both fMRI in data acquisition and data analysis of DINR fMRI. First of all, I hypothesize that the DINR signal consists of a deterministic neural response and noise, as well as a spontaneous ongoing neural activity. Unlike the bold signal, which consists of deterministic hemodynamic response and noise. When you accept this hypothesis, we may need a more effective way and a more optimal way of data acquisition and analysis for DINR fMRI. For example, in terms of data acquisition, we need to seriously consider how to make neurons more responsive to stimuli. For example, while minimizing their variation in coding adaptation, for example, by adding short rest periods between runs or focusing on the first 5 to 20 runs rather than acquiring many more runs. And in terms of data analysis, we need to consider how to separate deterministic responses from spontaneous ongoing activity. For example, in this case, for now is finding deterministic responses first by averaging across the subject and then returning to the individual subject to separate the spontaneous ongoing activity. Now I'd like to discuss the translation of DINR to human studies. First, let me briefly and roughly evaluate the possibility of a human DINR by only considering the number of neurons in a single bucket. When we consider the average a number of neurons and mass of the brain, neuronal mass density of the mouse is about 3 times larger than that of the human brain. And also we know that as the magnetic field strength increases, the signal sensitivity almost linearly increases. So the signal sensitivity in the mouse at 9.4 Tesla may be 10 times larger than that in the human at 3 Tesla. However, when we consider the single bucket size, for example, in the mouse, is 0.2 by 0.2 by 1 millimeter cube. In the human case, I saw 2 millimeter cube voxel size. The volume of a single voxel would be 200 times bigger in human images than in mouse images. So therefore, the signal sensitivity based on the neural density and field strength only, the sensitivity in human at 3 Tesla may be 20 times larger than that in the mouse at 9.4 Tesla. However, there are more things to consider in human DINR studies, like first, the reduced signal sensitivity due to much larger RF coil size. Now, another one is the reduced number of runs due to the limited scan time in human studies. And human studies also dealing with the awake case, not anesthetized. And also, In humans, the DINA study is dealing with quite different spatial scales. For example, the dimension of a single voxel in human is comparable to that of a S1 in mouse. But I'm going to show you our encouraging preliminary result of human DINA study at 3 Tesla now. We performed human DINA at 3 Tesla, repeatedly applying checkerboard patterns with 2 reversals during 500 milliseconds. And as a control, we applied the fixed cross pattern with a gray background. The TLNT was a 5 and 2 millisecond. Spatial resolution was ISO 3 millimeter. And the scan time was a 9 seconds per slice, and we applied 8 slices. So as a total scan time was 2 minutes and 32 seconds. And we measured the sequential dynamic response in the visual pathway, including LGN and V1 and V2. We selected and averaged activated voxels in each ROI from the DINA data itself using the criteria. In other words, we selected voxels with statistically significant activation compared with the baseline, which is the average of a pre-stimulation period. And also, we selected voxels having positive significance in more than 2 consecutive time points within 200 milliseconds. And this is the result of the sequential dynamic response in LGM and V1 and V2 compared with the controls, which reflects the neural activity propagation in human visual pathway. Finally, I'd like to acknowledge my great colleagues who contributed a lot to this study, as well as my research grant from the Brain Research Program of National Research Foundation of Korea. And also, I'd like to acknowledge my greater lab members. Thank you for your attention.